Thank you very much. Good afternoon, everybody. As already said, I'm going to talk about historical controls and when they might be used. If you have any questions, please answer them. Ask them, and I'll answer them to the best of my ability. If need be, quite happy to pause and take questions if they're burning or I haven't explained anything in sufficient detail, or if I go too fast, tell me to slow up. But I'm going to focus on historical controls. I'm going to start by saying a little bit about why historical controls are a subject matter of interest today. So a little bit of background to why I'm talking. And then some of the challenges as a statistician as I see the use of historical controls. And usually the two issues that you get from a statistical perspective is one is bias, which I will define so everybody has the same understanding. And the other one then is the inflation of the type 1 error or the increase in false positives that could be associated with historical controls. I'm then going to go on a little bit to say something about the benefits and how people usually argue for historical controls, which is quite often from the wrong angle, but I'm going to say a little bit about it and then explain why it's not necessarily appropriate. Some of the risks that you face when you actually look at using historical control, and then some case studies which have been successful where they've been deployed, and then make a few closing remarks. So why are historical controls of interest today? Well, they've been around for a long time and their use has been around for a long time. But in today's environment, regulators and the scientific community and patients are actually promoting innovation to speed up drug development. They want their drugs to the market faster. And there's significant use of and interest in real world data within clinical development these days. Primarily, I would say being pushed by sponsors, but also regulators are interested in it, patients are clearly interested in it. And this has come about particularly through the adoption of electronic health records. So it's much easier to get access to real world data as opposed to, let's call it, artificial clinical trial data. So one application of real world data that is under consideration at the moment is the use of that data as an external control to replace control arms in clinical studies. You know, most clinical trials that I actually design, or certainly towards phase 3, have a control arm in some way, shape or form. So the thinking is, why can't I just go out and get real world data and replace that control arm as an external control? The strongest arguments, as you'll see later in the talk, is when it's unethical or impractical to include a control arm in a trial. That's one of the ways that you can justify this, but it's not necessarily the most common justification, which I will share later, but that's really how they ought to be being used. So when you've got an unethical or impractical to get a control arm, go out and do this. So historical controls. For me as a statistician, there's really two major issues that I face when consulting about historical controls. The first one is the inflation of the type 1 error, and I'm going to assume most people are familiar with that. And that's about claiming a positive falsely. So in other words, there's a greater chance than the 5% that is usually accepted of actually claiming a success when actually there is no difference between a drug or it doesn't necessarily work. That's one area. The second area is bias. And the problem with historical control studies or using a historical control, it opens up the risk for biased treatment comparisons, much more so than in a controlled clinical trial that you would normally design. Okay. What do I mean by bias? Well, the simplest way and the best definition is probably given in ICHE 9, Statistical Principles for Clinical Trials. The principles are really about two things. One, minimizing bias, and the other is maximizing precision. But the definition that they give of bias is the systematic tendency of any factors associated with the design, conduct, and analysis and evaluation of the results of a clinical trial to make the estimate of a treatment effect deviate from its true value. So in other words, we set up a trial, we run a trial to try and estimate some kind of treatment effect, and we want to try and estimate the true value. But if we do things that design it inappropriately, we can deviate from that by design, by conduct. Bias introduced through deviations is usually in the conduct is usually referred to as operational bias. That's the terminology that's used. And then they lump everything else as statistical bias. So operational bias is deviations through the conduct. We as statisticians take everything else. Now, the problem with bias is it can occur in subtle or unknown ways, and its effect is not measurable. However, once included, it can seriously compromise the ability to draw valid conclusions from the clinical trial. I, as a statistician, can assess whether or not bias has been introduced, but I cannot adjust for it once bias has been introduced into a clinical trial design or operational conduct of a study. It's there and there's nothing that I can do about it. But that's really the definition of bias that I will refer to, something that actually makes the estimate of the treatment effect that you're interested in deviate from its true value. So where might bias occur? Now, I want to also acknowledge, I forgot to acknowledge on the first slide, that some of the material is taken from the paper, the use of external controls, certainly in the early stage, although the case studies are coming from a different source. So I wanted to at least acknowledge that. And this is one of the things I've just taken straight from that paper. So where can bias occur when you're using a historical control? Well, the first one that it can occur is in terms of the patients that are selected. You could have something called selection bias. The patients enrolled in the clinical trial are in some ways different to those patients in the historical control, which might be coming from patients treated in clinical practice. Now, there are mitigation strategies that you can get around the problem. So, for example, within a clinical trial, we have fairly strict inclusion-exclusion criteria based on what we want to do. Well, you could apply those same inclusion-exclusion criteria to the external control data that you might be using. You can also adjust for other issues using statistical methodology, such as propensity scoring, which matches patients on certain parameters, but you can never eliminate potential bias due to the patients being different. So the first source is potentially selection bias. Another one is calendar bias. Patients treated in the past do not necessarily align with those treated today. And the way to think of that is to consider standard of care. Standard of care evolves over time. So if you wanted to do a comparative study with standard of care and you're using patients from the past, what you might actually be doing is comparing versus a lower standard of care than is currently in operation today. So your treatment estimate would be different than the comparison versus the standard of care of today. That tends to be known as calendar bias, where over a period of time things change. So you can have calendar bias within your historical control study. One way to mitigate that is to take the control patients from the same calendar time as the clinical trial. So maybe using the real world evidence being collected today or being collected at the same time that you run the clinical trial, that eliminates some of the potential calendar bias that can exist. Doesn't eliminate it though, it minimizes it. Another source is regional bias. Patients' outcome can vary by regions. For example, carrying on the standard of care theme, the standard of care can actually vary by regions. So when you actually recruit your clinical trial, for example, you may recruit patients within US and Europe, but your real world data might come from right across the globe, which is US, Europe, Asia Pac, etc. And that can introduce regional biases because the patients from Asia Pac may be different in some way, shape or form than the patients in your clinical trial. So you can get regional biases that can occur. I'm not painting a very positive picture for historical controls, but I'll come back on when they can be used. But there are all these sorts of factors that need to be looked at. Another one is assessment bias. We, as trialists, as statisticians, as clinicians, we all understand that the gold standard is double-blind clinical trials where patients, investigators don't know what treatment's on, so that they cannot have any influence on the outcome assessment. That's what we all strive for. Well, the trouble with when you use historical control, you do know what treatment they're on. So you do know which ones are the new active. You do know, well, they know they're the historical control, although we can debate how much they know because it could be retrospective data. But you can have the risk of assessment bias coming into play. Now, again, a mitigation strategy is to try and choose objective endpoints. For example, in oncology, you might want to choose overall survival. For the more subjective endpoints, you might actually want to implement an independent review, similar to open label randomized studies, where even for the historical data, you review what the outcome is using the source data. But it's a risk. It's a potential source of bias that as a statistician, and I would even argue as a trialist, you need to be aware of when using historical control studies. It doesn't end there, I'm afraid. I've got some others that you can look at, all valid, all providing potential issues where you can, if you don't design it correctly, you can deviate from the true treatment effect. Another one is the endpoint bias, where, for example, how the endpoint is measured in your clinical trial can be different than in your routine clinical practice, which can introduce biases. One example is tumor images. How images are used in clinical research is different than how they're used for diagnosis and treatment within patients. So you've got what I would term different endpoint biases. Another one, if you're designing a clinical trial, I'm not sure what the right word here, but as statisticians, some refer to is immortal bias. When do you actually start the historical control patients off? It can be sometimes difficult, especially in the oncology area, to work out what the anchor for patients is. You know where the anchor is within a clinical trial, but let's suppose, for example, you're looking at early stage non-small cell lung cancer and you've got some historical data. When do you actually start them off for that comparison? Is it at the time of diagnosis? Is it a month after they've been diagnosed? Because normally within a clinical trial, they may or may not be recruited into the trial immediately, etc. But you can see how you can easily get potential biases by having this, what they call immortality bias, having different start dates between the clinical trial and the real world data. One way that to mitigate that is to clearly define the time point that you're interested in and ensure that they align and pre-specify it, but it doesn't eliminate it. Another one is study bias. Patients in clinical trials have different outcomes than in clinical practice due to placebo effects. You know, you go in to do a placebo controlled trial, you know that the placebo effect or whatever it is can be different in a clinical trial than it can be in real life practice. So again, you've got potential biases that can occur there. There are two others. One, variability between studies. It's not, you don't always get consistent treatment effects. So you can introduce bias by selecting potentially the studies that showed the worst outcome, which maximizes your treatment to be best. And there's something called intercurrent events, which I talked on last time, which is estimands. How they're handled within clinical trials can be very different than in real world data. So there's what, 8, 9 different sources of bias that are potentially of concern, certainly from a statistician, certainly for this statistician, and I would even argue for all trialists if you're going to go down the historical route option. Okay. Bias is probably the biggest problem because once it's in there, it can occur subtly that you don't know where it's occurring and you can't correct from a statistical perspective. The inflation of the type 1 error, which is the second one of concern for statisticians, I don't want to underplay it, it's still important. Usually there's two methods that we as statisticians operate when we look at historical controls. One is called static borrowing, where the external data is used irrespective of how it compares with the randomized trial data. So we've got this historical control data that we're going to use. That's called static borrowing. Another one is dynamic borrowing, where we only use if it's consistent with the randomized trial data. So we do some checking and we have to ensure some kind of consistency. That latter one reduces the bias going on the last one, but it's still there. Now, those two methods are available. They're quite well published in the literature, different approaches. I'll say a little bit about them in a minute, but I would say the jury is still out in terms of the actual inflation that can occur to the alpha level, and research is still ongoing. If you want a very crude breakdown, in terms of the static borrowing, it tends to come under a frequentist type of approach, your standard hypothesis testing, and the standard method tends to be something like propensity scoring, where what you need is individual level data in terms of the historical data, and you can then match that, the historical and the clinical trial, using various pre-specified prognostic covariate factors, and that's something called propensity scoring, which is commonly used. If you want to use a Bayesian, you tend to go for a mixed prior type of approach. The best way is probably illustrated here, which I borrowed from a GSK slide. As an example, let's suppose that you're interested in a pediatric drug development and you've got some information from an adult development program. That's your historical information. So if you look at it in the top left, I've got information about the adult data, which is the historical information which I can use. However, I don't know whether or not it actually is consistent with the pediatric. So what I might do is have that prior information, that prior knowledge from the adult data. I would also have something else called a skeptical prior, assuming
Sorry, Alan, can you hear me? I don't know if it's your...
I can hear you.
Okay, you're going in and out for me, but maybe that was me and not everyone else. I'll just let you go ahead. You're co-host anyway. If for some reason my my connection isn't good, I don't know if that was your connection or mine, but go ahead. Sorry.
Can other people hear?
I'm not seeing anything in the chat.
I'll assume yes, unless I hear otherwise.
Was in and out, back now. I've just had someone say. So I think maybe you might have gone in and out, but anyway, it seems to be back now.
How far do I need to go back?
I think, well, for me, you were just talking about probability here in the left-hand corner, just combining the two.
I'll start from there. I apologize for going in and out. I guess that's technology that I can't control. So you might have some information on adult data. That's one piece of historical information that you have. You might have no information. You might assume I know nothing about the pediatrics. And what you want to do is combine them in some way with a weight W, in some way W in 1 minus W. And the more the adult is consistent with the pediatric data, you're going to increase W. But if the adult data conflicts with the pediatric data, you want to downweight the adult data and just assume the skeptical prior. So what you're doing is borrow information in a dynamic way of combining your historical control, assuming nothing. I'm not going to go into how the maths work, but that's broadly it. Okay. In terms of the benefits, there are benefits of using historical control, and I've already mentioned very early on that the benefits really come in terms of when you've got rare diseases, or it's difficult or unethical to actually recruit a control group. But often what I see is quite a lot of sponsors come along and say, oh, we use a historical control because it reduces time and cost. But the trouble is, quite often they're overstated. Even these are overstated, and is not the right way to justify a historical control. So for example, one way that you could do it is potentially think of it, well, I can eliminate the control group. If I've got real world data, I only need to recruit the active group. So I've saved myself all the costs associated with and time associated with recruiting the control patients. Firstly, it isn't a zero cost gain, the real world data. You've got to invest in getting it. You've got to invest in ensuring the quality of it. You've got to invest in making sure you've got patients. So it's not a zero cost. The other one is you don't actually reduce the sample size. So you don't reduce the time and you don't reduce the cost. In fact, the sample size can go up because in a single arm study, to allow for increased heterogeneity, sometimes what you have to do is increase the sample size. So you're certainly not reducing costs by 50%, just eliminating the control patients. You've probably got to recruit more patients in the active study. The other thing in terms of time, if you've got a large study, companies, sponsors tend to open more centers. So, you know, taking it to a simplistic, let's suppose we've got a 400 patient study, we're going to open up X centers. Well, for a 200 patient trial, I'm going to do X over 2. But you don't always achieve that time saving because you don't always know which is the X over 2 centers to recruit from. And it could be you choose the wrong ones and it takes you longer. So the arguments based on time and cost are really the wrong direction. The strongest case for using a historical control trial is based on ethical arguments, and that's really where you want to go. So for example, in pediatric patients that I just did, you might be able to say, well, it's actually unethical to expose too many children. So therefore, what I'm going to do is I'm going to use historical information from the adults, but that's where you have to take the totality of the development program. Another one might be rare diseases. You know, it could be ethically, it's a challenge for the rare diseases, etc. So that's another case where you might be able to use historical controls. In terms of the risk, we've already touched on some of these in terms of the bias. It's about getting appropriate historical data that matches in terms of the patient population of interest, in terms of minimizing calendar bias, minimizing assessment bias. So one of the risks you really have to focus in is making sure the fit of the external control is current in the current treatment context. If you don't, it's a huge risk to the development program. And actually that can result not only in bias, as I've talked about, but potentially you can draw the wrong conclusions. You can make false investments. So quite often in early phase trials where historical controls can be used, if you use the wrong control group, you can actually conclude that your drug is much better than something that will never get to market. And you can do investments and eventually it will catch up with you when you do the control groups that you've gone down the wrong path, which can also result in delays because you then have to correct it. On the other hand, if you get the wrong treatment groups or the wrong comparator, it could be that you kill a successful drug as well. So there are significant risks in terms of when you deploy them. The other advice I'd give you is always think that the historical control data should be a comparable quality as the study data. So don't think that just because you're using real world data, historical comparison, I can actually live with a lesser standard for that data. You can't. Essentially, you have to ensure that the quality of that data would match what you would have got in a clinical trial. So all these issues that I've talked about have to be well thought through, documented, and presented, certainly in the environment I am, to a regulatory body in order to accept a historical control study. Right, I've given you the bad news. So where are companies trying to use them? Where might you use them? So one company that I'm aware of is something called Unlearn. Now, I have no vested interest in Unlearn. I just came across this, and I like the notation of the twintelligent concept that they're promoting, whereas what they're trying to do is try and identify the historical twin for the patient that's recruited within a clinical trial. And they put that their methodology is patented, and they submitted it to the European agencies, the FDA agencies, to say, will you accept this as an appropriate method? They put in 3 case studies, and not surprisingly, the authorities didn't come back with an overwhelming yes. They basically came back, and I highlighted some of the extracts from theirs that said, you know, if you've got a model that predicts the outcome in a future clinical trial and you've proven that model, then to some extent that can be used as controls instead of recruiting patients into a clinical trial. So as long as you've got that historical data for which you've done that modeling, then they may accept it. They didn't give a blanket, because obviously they said, well, the model can vary depending on the disease and the indication, but where you can and the outcome is fairly predictable, yes, we're willing to consider this. And I'll give a case study, a more specific case study, when that might be applied. I think, and you need to go back and look at this, where they were trying to get to was in Alzheimer's. You know, Alzheimer's are very long-term studies, involve lots of number of patients. So they were trying to come up with a model that predicted the deterioration in Alzheimer's, which they could use instead of a control group within an Alzheimer's study. So that the authorities were somewhat positive in selected cases where you can develop a predicted model. Another one in oncology, there's this brochure on Friends of Cancer, and what they do is they talk about the different scenarios where you might have an external control, starting from uncontrolled studies, single treatment studies. The reason why I put it in is because, again, the regulatory perspective, they say that you can do it in limited circumstances, which again, it is consistent with this message. And also you've got to have a careful assessment whether external controls will scientifically be feasible based on the outcome. So it's not just a case of I'm going to do it, you've got to justify it. Some specific cases, metadata, they're a big data management, they're the people who own the remote data management, Oracle, it's not Oracle Clinical, but that metadata is where we use for collecting data, for those who don't know. They went through a PR exercise where they mentioned that they got the FDA agree to use a trial design using their existing metadata that they have from other studies in recurrent glioblastoma, which is a disease with poor prognosis and higher medical need. And with the sponsor, they went to the FDA and said, look, we don't want to use a control group within the study. What we want to do is propose a historical control group. They had discussions with the FDA, and where that design came out was that they had a 1-to-2 in terms of control to historical control. So one for every 1 patient in the study, they were allowed to use 2 historical control patients as the comparator. I haven't seen the results of that trial, but it's an example where the FDA said, yes, we can use historical data to some extent. Another one that I'm aware of is in cystic fibrosis. I said that one of the areas that the Unlearn came out was where you could develop a model. Well, within cystic fibrosis, one of the endpoints is, and I'm not an expert in cystic fibrosis, is annual rate of decline in percentage predicted FEV1. And they developed a model. If you look at the curve at the bottom, the red group was the control, the historical control, where they managed to show that it was a fairly predicted decline over a 2-year period. in FEV1 using historical data. They did a single arm treatment where, in a sense, they managed to show that their decline, well, they improved to start with, and the decline was slightly less, and that was accepted in terms of the work that they've done. So that's one case of an example where a model had been developed and they used that model to do the prediction and just reduce the number of patients. Another one is Brineura. Again, these are all in the public domain, so you can go and look at this. And the FDA approved it for the treatment of Batten's disease, or a common form of Batten's disease. They used a historical control population. They were actually initially looking at 4 domains, 4 assessments, one on motor, one on language, one on visual, and one on seizure. But only 2 domains were measured in the historical control. So they only had motor and language available for which to do their historical control, despite them wanting to look at 4. There were also several differences between the historical control population relative to the trial population. But in a sense, the FDA and statistical, the FDA, the stats reviewers didn't like the language domain, so they threw it out, and they did an analysis focused on the motor domain of the historical data versus the active data. They analyzed it to start with at week 48, and they said no, that the findings were inconclusive. And I haven't managed to track down completely, but I assume that's because the potential biases were not, that the treatment effect didn't outweigh the potential biases. But the FDA, the agency, came back and said, oh, we'd like you to do further analyses at 72 weeks and 96 weeks. And they changed how they were actually matching the control patients. And based on that, they did see larger differences at the later time points. With these matched, they saw, I think it was 29 and 59% at those later time points, and they got approval for it. So it's an example where historical control can be used. My last one is Blincyto, which is some form of lymphoblastic leukemia. The company ran a 185 patient study. It was a single arm study, and they wanted to compare the information with an historical cohort based on European national study groups. They had a couple of analytical approaches. One was, the main one that they went for eventually was propensity scoring, which I mentioned is a recognized technique. There were differences between the patients in terms of pre-treated, so they had to be adjusted, and there were all sorts of different ways of doing it. But essentially, the results showed this for the different adjustments, and you can see from the curve that there was a benefit in terms of the drug relative to the historical control data set. So that was successful as well. Okay, I'm wary of time. I said I'd talk for about 30 minutes, 35 minutes. So I'll make a few closing remarks and then take questions. From this statistician's perspective, randomized clinical trials, I think, will remain the gold standard to ensure robust data generation for the foreseeable future. I think historical controls can be used, and there are cases that you can quote, but if you're going to go down that route, you need to use them based on ethical arguments, not costing time. So the obvious places for me are things like rare diseases, pediatric diseases that are life-threatening or serious. So you need to make sure the cases are well thought through. And we as sponsors tend to, let's say, try and push as much as we can, but that doesn't necessarily help us as the industry. Any treatment effect that you're trying to detect or observe must exceed any biases that could exist with the historical control. And I gave a whole list of potential biases that can exist, which need to be thought through and documented and discussed when proposing a historical control. So you need to understand when they could occur and what the impact could be. The external data needs to be of high quality. You can't get away with substandard data, so it's got to be thought of akin to clinical trial data, but each of them, when you're using historical controls, it's going to be argued on a case-by-case basis. It's not a panacea. It's not going to suddenly say, oh, I can save half my patients. I've got some patients out there. It's not a panacea. It has to be argued on a case-by-case basis, primarily on ethical considerations. I probably think the metadata is probably where we will settle where possible. So in other words, where you've got a combination of control patients within the clinical trial supplemented by a historical control. And the reason for that is you can check consistency between your control patients and your historical control patients easier. So I quite like that. And I suspect that might be a place where the industry, the regulators settle, certainly in the short term. What the ratio is, well, as I said, they went with a 2-to-1 ratio. I haven't seen any statistical justification or clinical justification for that. It just seems a right number, but something like that might be the way to go. But regardless, you've still got to get good quality external data. Okay. And with that, can I thank you for listening? If you want the paper for some of the earlier stuff, it's this, and it does involve people involved in it were from the regulators as well as industry. But the case studies won't be in there. Those are coming from a different source. Okay. With that, can I thank you?